Thanks to the organisers for this invitation. I'm very flattered to be invited to give you this talk. I thought long and hard about what to talk about today, and I thought I would use this opportunity to deal with a few of the dogmas that have arisen in the field. So I'm going to start off by talking about Trimethyl K4, and then I'm going to introduce you to the yeast metabolic cycle and how some of the misconceptions about how this modification is regulated have come about. I just want to acknowledge the people in the group who've done the work I'm going to talk about today, including David, who's going to be talking later in the session. He was a postdoc a long time ago in my group when some of this work was done. So this is a summary of what I'm going to talk about today. So first of all, I'm going to show you that acetylation of lysines can influence local protein structure. And I'm going to show you that acetylation at lysine 14 is associated with isomerization of the A15P16 peptidyl proline bond and makes it bend and go into cis. And this influences the deposition of trimethyl K4. So that's a mark on the same tail but a long way away. And lysine 14 and P16 are required for optimal trimethyl K4. And these features influence the balance between methylation by the SET1 complex and SPP1 and demethylation by JHD2. What I'm then going to show you is that different genes respond differently to this regulation. So trimethyl K4 in these mutant strains, and also in a strain lacking SPP1, the levels remain high at the RPGs and RIBI genes, so the genes involved in growth and ribosome biosynthesis. And most of the marks are lost from the environmental stress response gene. So you don't get a universal loss of the modifications, a stratification. And the classes that are most and least dependent on lysine 14 and proline 16 are the genes which are associated with quiescence repair, which are the environmental stress response genes, growth and synthesis, which are the RPGs and RIBIs. And these are genes that cycle in the yeast metabolic cycle, and I'll introduce this to you. And then I will show you that in fact, when we're talking about, at least in yeast, concepts of gene activation and repression, probably what we're talking about is very simply a change in the number of cells in the population that are in one of these two phases. So that's what I'm going to talk about today. So I'm going to start by introducing Trimethyl K4 to you. Many of the people in this audience, including Francis, who had one of the first papers on the SET1 complex, have worked on this field. And we know an awful lot about this modification, but it's still a little bit of an enigma, certainly in yeast. It's not an essential modification. The question is what it does. So trimethyl K4 is a near universal modification which is present on active or potentially active genes, including bivalent genes in embryonic stem cells. It has a distinct distribution, often enriched around the +1 nucleosome and often co-occupying a nucleosome with a variant histone H2AZ or HTZ. So here's a distribution in a gene which is expressed in embryonic stem cells, and it's very like the sort of distribution you would see in yeast. We know from lots and lots of very nice experiments that the modification recruits a wide variety of chromatin modifying complexes and transcription factors, which are involved in initiation, post-initiation, and splicing, so all aspects of RNA production and metabolism, and that it acts positively and negatively to influence transcription. So it recruits things like ISY in mammalian cells, ISY in yeast, lysine acetyl transferases, components of TF2D, SPP1, one of the components of its own complex here, which I'll talk about in a minute, CHD1, NERD, etc., via domains such as the chromodomain and PHD. In yeast, certainly, levels of trimethyl K4 generally correlate with levels of transcript, so there's a correlation with the amount of gene expression product. But paradoxically, much of the work in yeast suggests that trimethyl K4, although it's a mark of active or potentially active genes, is often associated with the repression of genes. In metazoans, there are a number of methyl transferases for trimethyl K4. In yeast, there's one complex, a SET1 complex. The modification is deposited by SET1. And one component of the complex, SPP1, which contains a PHD finger and actually binds to trimethyl K4, is one of the components of the complex which is actually required for trimethyl K4. And a yeast strain in which this SPP1 component is deleted loses a good amount of its trimethyl K4, but you'll notice it doesn't lose all of it. And we made this observation a long time ago with Tony and other work that we were doing very early on in this. And I've always wondered what that residual level of trimethyl K4 means, what's actually going on. So the questions that we started this story with is the question of, does the deletion of SPP1 from the set1 complex influence trimethyl K4 just generally, so the levels go down at all genes in the yeast genome, or does it affect different genes? And you know I'm going to tell you it affects different genes. And the other question we wanted to know was whether there are other mutations that have the same effect as loss of SPP1. So I'm going to start off with the second question. So actually, we and others had shown a long time ago that a substitution of lysine 14, an alanine substitution, also results in loss of trimethyl K4. And if you quantitate a number of Western blots, they're quite hard to quantitate, as anyone will know, you can see that the lysine 14 substitution here and the SPP1 have very similar residual levels. We decided to look at the other residues around lysine 14 on histone H3 to see if any other residues have the same effect. And we discovered that proline 16, although the effect's not quite as marked as with SPP1 or lysine 14, also affects the levels of trimethyl K4 globally. So these are Western blots. It affects about 50% of the global levels. And you get a similar effect with a valine substitution, which is about the same size as a proline or an alanine substitution. So an SPP1 delete, a 14A substitution, or a 16V or A substitutions affect global levels of trimethyl K4. So the first question I addressed is this effect the same at all genes? And this is just a chip PCR experiment which shows you very clearly that it's not. So here we're looking at levels of trimethyl K4 relative to wild type at 6 genes. So we've got 2 ribosomal protein genes here and then a series of other genes that we just happen to have PCR primers for, FMP27, SEN1. These are long genes, PGK1 and PDC1. And you can see relative to the wild type, both the K14A and the SPP1 delete tend not to lose much of their levels of trimethyl K4 at these genes, whereas at these genes you can see the levels are markedly reduced. So one explanation for the residual levels in the global Western blots is the residual trimethyl K4, which is present on these genes here. And I'll show you in the genome-wide analysis that is in fact the case. Okay, so the next question we wanted to know was, I've shown you what an alanine substitution at lysine 14 does. Are all substitutions at lysine 14 going to have the same effect? And the answer is up here, and it shows you very clearly that they don't. Now, one assumption that has been made right from the word go was that because the 14A substitution could not be acetylated, that acetylation of that residue was what was required for trimethyl K4. And well, this result shows you that that can't be the case, and I'll actually show you in more detail why that can't be the case. So if we substitute 14 with R and Q, you can see we have very different effects. So the R substitution here has much less of an effect on either the global levels or the levels at individual genes compared to the 14A or 14Q. So that does suggest that it can't be acetylation. This just shows you that the 14R substitution in a wide range of different strain backgrounds has the same effect. So it's not a strain-specific effect. It happens in a lot of different strains. And that you see this same, this relatively high residual level at all these genes compared to the 14A strain. So we don't think it's acetylation. And here's another bit of evidence which also suggests that it's not acetylation of lysine 14, which is helping to deposit trimethyl K4. So here what we've done is a global Western blot in a strain with a 14R substitution. So the 14R substitution cannot be acetylated. If you then subsequently knock out either GCN5, which is an acetyltransferase for lysine 14, or ADA2, which is a component of the SAGA complex, plus the NUA3 acetyltransferase SAS3, so this knocks out 2 acetyltransferases, they affect global levels of trimethyl K4. And this is part of the evidence that contributed to this idea that it was acetylation by the acetyltransferases which was required for global levels of trimethyl K4. But if you have a look in the 14R strain, which has relatively high residual levels, and then you knock out GCN5 or ADA2 and SAS3 in these strains, you can see they still lose global trimethyl K4. And that means that what these enzymes are doing when you knock them out cannot be lack of acetylation at lysine 14. There's another explanation for why these strains are losing global levels. So the effect of knocking out the acetyltransferases or the substitutions, certainly for the acetyltransferases, their effects on levels of trimethyl K4 are likely to be indirect. And I'll come back later and show you what actually we think is going on there. So lysine 14 acetylation is not what's responsible for global levels of trimethyl K4 in the 14 substitution strains. So we were interested, because 14 and 16 are only 2 amino acids apart, we were interested in whether that they might be doing something along the same lines, working on the same pathway. And so this again is showing you H3 trimethyl K4 levels at the individual genes shown here. So we've got a ribosomal protein gene, a couple of long genes, and PGK1. Here are the wild type levels. And here you can see high levels at RPS15 and the low levels at the other 3 genes in 14A. 14R high levels. So what we then did to ask whether lysine 14 and proline 16 were somehow talking to one another was to make double substitutions. And the track I want you to concentrate on here is, if you have a look at the 14R strain, as I've told you already, this has high residual levels at all genes. If we combine the 14R mutation with a 16V mutation, you can see we have quite a severe effect on both the levels of the ribosomal protein gene, but particularly on the levels of these proteins here. So this suggests that the substitution at 16V modulates the effect of the 14R substitution on the global levels. So the question is, what's going on? So that's what I'm going to try and talk about today. So I mentioned to you earlier that prolines, the peptidyl-prolyl bond, undergoes cis-trans isomerization. And there are a group of enzymes called peptidyl-prolyl isomerases which can speed up this isomerization, but prolines will isomerize naturally in proteins or peptides. And in a peptide, about 90 to 95% of the peptidyl-prolyl bond will be in trans and about 5 to 10% in cis, and there'll be a slow isomerization in a peptide. So we can sort of envisage what the tail of histone H3 might look like when proline 16 is in trans, so this is when it's effectively straight, or when it's in cis, and this is when it's bent. So it's about a 90-degree bend that it introduces into the histone H3 tail. And you can see that this is going to influence quite markedly the relative position of the residues down this end of the tail compared to when it's like this. I mean, I can't possibly draw you a higher order structure, but you can imagine you've got 2 nucleosomes next to each other, you've got 2 H3 tails on the same nucleosome. The way that these residues are presented will be different. Okay, so the first thing we wanted to do was to try and see if we could find any evidence that prolines underwent conformational change in the cell. And what we tried to do was to raise antibodies to the cis or the trans state of proline 16 by hydroxylating the proline ring. And by hydroxylating it, you can fix a cis and a trans state. Well, that's what the chemists tell me they do anyway, and that's what they said they'd done. And they gave us these peptides, and we raised antibodies, and it required a huge amount of selection. In fact, David and Matthew Sales did this work here to produce a very small amount of an antibody which could specifically recognize the cis-16 against the trans, and then an antibody which could recognize the trans against the cis, and also the unmodified, because most of the unmodified will also be in the trans configuration. And then we use these antibodies on Western blots and also on chip qPCR. And what you can see here is that here's the trans antibody, which tends to pull down chromatin towards the 3' end of the gene. I'm not going to talk any more about this. But this is the cis antibody here, and this produced a high signal at the 5' end of this long gene. It's 8 kb long. And interestingly, its distribution was very similar to the distribution of Trimethyl K4. So in vivo, we were able to show that 16 in cis, so the bent form, is found at the beginning of this gene here with a similar distribution to Trimethyl K4. So just bear that in mind. Bent correlates with Trimethyl K4. So then we tried to do some biochemistry to ask whether we could show on peptides that the proline adopts alternative configurations. So this is a very busy slide, I apologize. So this is the bromodomain here. Bromodomains bind to acetylated lysines. And the test that we're using here is the binding of the bromodomain from SPT7, which is actually a component of SAGA, but that's actually not relevant here. The SPT7 bromodomain we discovered in separate work interacts with lysine 18 on histone H3 when it's acetylated. And you can see here, here's a peptide, the peptide's 11 to 26 here. When it's acetylated at lysine 18, it binds to the SPT7 bromodomain. This is SPR data, and this is pull-down data. You can see that there. So what we then did was to introduce other modifications and substitutions in this peptide to ask how they influence the binding. So the first thing we did was to substitute proline 16. That's what we're interested in. And we substituted it with a valine so we could fix it in the straight conformation. And when we do that, you can see that it actually improves the binding. So this is increased binding here on the SPR, and you can see a small increase here. So having the 16 in trans in the straight configuration improves the binding of the bromodomain to lysine 18 acetylation. And then we started introducing modifications or substitutions at lysine 14 to see how lysine 14 might affect proline 16. So when we acetylated lysine 14, so we have a doubly acetylated peptide, we also see increased binding. You can see that over here and here. So this is quantitative data from about 20 different experiments. Interestingly, when we introduced the arginine substitution at lysine 14, remember this is a substitution which doesn't really have much effect on global levels of trimethyl K4, you can see that it virtually abolishes binding. So binding is abolished by a 14R substitution. So I'm sure some of you are thinking, oh, it's just the charge or whatever, you know, it's just affecting the binding of the protein. But the key experiment, I think, here is when we took this peptide here and introduced a valine 16 substitution. So we have the 14R and now the valine 16. We restore binding, and binding is restored to better than wild type levels. So one conclusion we drew from this is that the state of lysine 14, so whether it's acetylated or has, well, I didn't show you this data, but a glutamine or an alanine substitution, the alanine substitution doesn't have much effect, influences the conformation of P16 and tends to make P16 adopt the trans configuration and improves the ability of the SPT7 bromodomain to bind to the peptide. So SPT7 likes to bind when proline 16 is straight, and these substitutions at lysine 14 affect that. The 14R substitution here, though, is doing the opposite. We think the 14R substitution is promoting the bent conformation of the tail, and that abolishes the capacity of the SPT7 bromodomain to bind. And as I said, when we re-straighten out the tail with a valine substitution to make it straight again, it binds. So it's not an indirect effect of charge. So that's one piece of evidence that the proline can adopt different conformations, and this can functionally affect the way proteins bind to this tail. This is a much more direct assay for the effect of a modification or substitutions at lysine 14. So this is a classic assay for looking for proline isomerization. You synthesize peptides, which are hard to synthesize, which contain a little bit of histone H3 here. Here's the proline 16, there's lysine 14. We then have a phenylalanine and a paranitroalanine residue. And this phenylalanine PNA bond can be cleaved by chymotrypsin, but chymotrypsin will only cleave this bond when it's in trans, when it's straight. It won't cleave it when it's in cis. And so you can measure the rate of PNA release from the peptide, and we measure this initial rate over the first few seconds of the reaction. And the larger the bar here, the faster the rate of cleavage, and the higher the trans content of the peptide. And I should say that the chymotrypsin cleavage is much faster than the natural rate of isomerization of the proline. So what you can see here is that the unmodified peptide has a certain rate. When we acetylate the peptide, and our prediction is that acetylation will make it go straight, it is cleaved faster. A 14Q substitution does the same thing. A 16V, still a bit faster, that should also be straight, but it's going to influence the environment here. But importantly, the 14R substitution has a slower rate than wild type, and what we're proposing is that the 14R makes this bond more likely to go into cis. So we can summarize this first part by saying that we think that the prolines in the histone H3 tail can adopt alternative conformations, and the state of lysine 14 can influence this. So when 14 is acetylated, we think the bond tends to be in the trans configuration. When we have an arginine substitution here, which is a little bit bulkier than the normal lysine, it tends to adopt the cis-bent conformation. And our prediction is that in vivo, we have transitioning between the two states, which reflects dynamic acetylation at lysine 14, so 14 becoming acetylated and deacetylated, and this is then going to affect the amount of trimethyl K4 on the end of the tail. So we think that the modifications at 14 affect trimethyl K4 by affecting the conformation of the tail. And this is just a little bit of data. We've got a lot of data to support this, which suggests how this alternate structure might be working. So there's two bits of data here. On this side here is the binding of SPP1. So SPP1, remember, is the component of the set1 complex, which is specifically required for trimethyl K4. And here we're looking at the binding of SPP1 to our 6 genes here relative to wild type. So what you can see is that in the 14A substitution, the 14Q substitution, and the 16V substitution, all of which are associated with the trans straight conformation, the binding of SPP1 is reduced. It's not we don't see exactly the same effect in all genes, which is not really surprising. But the 14R has binding as good or sometimes better than wild type. So that might explain partly why the 14R strain retains high residual levels of trimethyl K4, whereas these strains tend to lose more. But that's not the only contributor to this. We also see an effect on demethylation. So JHD2 is the demethylase for trimethyl K4. And in fact, when we substitute any of the residues at lysine 14, so here's an example with a 14R or a 14Q substitution, we're overexpressing JHD2 in this strain from a gal-inducible promoter. And as we induce expression, you can begin to see an increase in demethylation of trimethyl K4 in the wild type, which is coincident with increased expression of JHD2, which is HA tagged here. When we have the 14 substitutions, a 14R or a 14Q, we tend to see a faster rate of demethylation. So it suggests that the final level of trimethyl K4 is a balance between deposition by the set1 complex and SPP1 and demethylation by JHD2. And what the 14 substitutions do by altering the conformation of the tail is to change the balance of acetylation and methylation and demethylation. So the 14A, Q, and 16 substitutions swing everything in the favour of just the demethylated form, because you get less set1 bounds and you get increased demethylation. Whereas the 14R has much more wild type levels because it is able to retain relatively high levels of the set1 complex, although has a higher level of demethylation, so the balance is more in favour of the wild type conformation there. So we think it's a balance between methylation and demethylation, which is perturbed in these substitutions, and this is because the conformation of the tail is changed. Okay, so we were interested in how the 14A and 16V substitutions are affecting levels of trimethylation on lysine 4 genome-wide. So we did a genome-wide ChIP-seq in both the 14A and the 16V strain. And what we discovered was that we discovered what I've already shown you, really, that there's a gradation in the amount of trimethyl K4 which is lost. So at the top, the lysine 14 and P16 independent genes, so the genes which lose least or have highest residual levels in the K14 or P16 substituted strains, are the RPGs and RIBs. I've already shown you some of that data, but this is now the genome-wide data. Whereas the genes which showed most dependence on the integrity of lysine 14 and proline 16 for trimethyl K4 are the SAGA-regulated genes, which are incidentally also the environmental stress response genes. And you can see the data here. So in the black line here, I'm sorry this is not very clear, the black line here is all genes, so this is the pattern for the 14A classes and the 16A classes in the wild type. And then in the sort of middle grey, which is this line here, you can see the K14 and P16 independent genes, and they have quite high levels similar to wild type. Whereas the K14, P16 dependent genes, they have trimethyl K4, but the levels are lower. So it's worth bearing in mind these genes here, these SAGA-regulated genes, generally have less trimethyl K4. But what you can see is the effect of the 14A or the 16V substitution here. So whereas all genes lose a lot, as I've already told you, they still have some residual levels, the independent class retain quite a lot, and the dependent class go down to virtually nothing. So in fact, these genes here lose most, and these genes here lose least. And you can see a similar distribution here. So we can stratify the genes in terms of, some that lose the modification and some that don't, depending on which class they fall into. Okay, so I'm going to summarize what I've told you so far, and then I'm going to explore why and explore a little bit about what's going on. So what I've shown you so far is that there's plasticity in control of the levels of trimethyl K4. And we think this occurs via signaling to lysine 14. So lysine 14, a lot of signal transduction pathways will affect acetylation at lysine 14, and that then affects isomerization at proline 16, and this will determine the rate of set1 deposition of trimethyl K4 and JHD2 dependent demethylation, and set the residual levels of this modification. So we think it's much more dynamic than we might already thought. We think that acetylation at lysine 14 favours the trans conformation, and this actually reduces the global levels of trimethyl K4, which is exactly the opposite of what the literature will tell you. The literature will tell you that lysine 14 acetylation is required for trimethyl K4, and I'll come back to this later on. So the ribosomal protein genes and RIBs, these maintain high levels of trimethyl K4 independent of lysine 14, proline 16 and SPP1. I haven't shown you all the data for SPP1. Whereas environmental stress-induced genes are most sensitive, and they lose most of this modification when the tail is compromised. So the question is why? And we still haven't answered this question fully, but I'll give you a little bit of an explanation. So when we were stratifying the global effects, we noticed that our lysine 14 and proline 16 independent genes were highly enriched. I haven't got the significance values here, but they're very highly enriched in genes which cycle and peak in what's known as the oxidative phase of the yeast metabolic cycle, whereas our 14 and 16 dependent genes peaked at a different phase of this metabolic cycle in the so-called reductive charging phase. So before I come back to explain what might be going on, let me just introduce you to the yeast metabolic cycle. So yeast, like all cells, has a metabolic cycle. So this is not a yeast-specific effect. It's just easier to study in yeast. So rhythms and cycles are what makes up cells. And in fact, over half the genome, in fact, there's a new paper just coming out identifying another class of cycling genes. 3,500 at least genes in the yeast genome cycle during the yeast metabolic cycle. So their transcription levels go up and down, and I'll show you some profiles in a minute. So the metabolic cycle basically divides up metabolism into two phases. So there's a phase in which oxygen is used, in which ribosomes are synthesized, amino acids are synthesized, and sulfur metabolism occurs. And this is associated with growth and synthesis. So this is a growth and synthesis phase. The metabolic cycle can occur independently of the cell cycle, so the cells don't have to be actually dividing. So if they were dividing, it would be growth. If they weren't, it would just be a phase of synthesizing and making good. That's then followed by a phase where if cell division is going to occur, the cell division cycle occurs and mitochondrial biogenesis, so the mitochondria also divide. And then there's a very important phase, which is the reductive charging phase here, where things like fatty acid oxidation and glycolysis occur, high levels of autophagy, and effectively you can think of it as a phase of quiescence and repair. So here the cells are repairing damage, and they're getting ready, synthesizing acetyl-CoA and NADH in order to grow again. So the yeast go through these cycles. So what does this mean? Why do these genes peak in the oxidative phase? Well, you know, they're ribosomal, they're ribby genes, so you expect they would. And why do these genes peak here? So these are actually profiles of cycling genes. So the yeast metabolic cycle was originally defined in Steve McKnight's lab, and Benjamin Chu and the Botstein lab have been instrumental in our understanding of this. But this is data from synchronized cycling populations. And what we're doing here is looking at a slightly different experiment, which I'll come back to. So the question we're trying to address is, why do our 14 and 16 independent genes peak here, and these genes correlate with genes that peak in the reductive charging cycle? So these are the cycles for oxidative and reductive charging genes. We were doing a separate experiment in yeast where we were doing what I think is a really simple experiment, which is to take exponentially growing cells in glucose and transfer them to galactose and induce them for 3 hours and ask what happens. And we were using a technique called NetSeq, which measures the RNA in the active site of the polymerase. So we were measuring transcription. I'm not going to go into the details here. But what we discovered was that when you do this glucose to galactose shift, we also saw quite an interesting stratification of the growth genes, so the oxidative phase genes, which become repressed when you shift cells from glucose to galactose. And you can see here there are 309 genes which show a greater than 3-fold decrease in transcription. This is not transcript levels, but transcription when you shift the culture from glucose to galactose. And you can see that these are genes which would normally cycle in the oxidative phase of the metabolic cycle. Whereas the same shift, you start to see an increase in your reductive charging phase genes. So this is the key over here. So these are the genes that increase. So what we're doing here is just changing the carbon source of the yeast, and we're changing dramatically the classes of genes which are upregulated and downregulated. Now these genes have nothing to do with glucose repression, or most of them don't have anything to do with glucose repression or galactose induction. Obviously a few of them do, but most of them don't. So the question is, what's going on here? What might be happening? Just one other aside here is that many genes in yeast cluster, And this shows you net-seq profiles. So this is the transcription profiles and the steady-state RNA profiles for a cluster of yeast genes here. This is one of the genes that we've studied quite a lot. It's a gene called HMS2. And the reason that I'm introducing it to you here is because it has an antisense transcript. And this is relevant to the story I'm going to tell you. So here's the sense transcript here, which is induced in glucose and repressed in galactose. And the antisense is induced in galactose and more repressed in glucose. And you can see that the transcription is reflected quite nicely in the steady-state RNA levels. And incidentally, just a few take-homes here, you'll notice how much overlapping transcription there is in yeast. So these transcription profiles go way beyond the extent of the transcripts. And, well, in a separate piece of work, we've shown that this gene here mediates transcriptional interference of its gene downstream, and that's part of the mechanism which regulates the cycling, but that's actually not the point of showing you this. The point of showing you this is that if we do single molecule RNA fish on HMS2, this gene, and its antisense, I don't know whether you can see this, spots before the eyes, but here's a field view of cells and here's a few separate cells. So this is single molecule RNA fish where we're using probes which are labelled with fluorophores to detect both the sense and the antisense transcript for this gene in the same population of cells. And what you can see here is that generally the sense and the antisense transcripts are generally present in different cells. Okay? So a cell will either contain the sense or the antisense, or if it contains both, it's generally got a site of nascent transcription in the nucleus and is switching from one to the other. And with this gene, I've shown you already that levels of the sense transcript decrease in glucose and levels of the antisense transcript increase in glucose. If we quantify what happens to the distribution of transcripts in the cells in the population, you can see in glucose, the cells contain more sense transcript than antisense. No, I need to start again and say that differently. Cells contain either sense or antisense, okay? So the number of cells containing sense is large, over 60%, and the number of cells containing antisense is small here in glucose. And when we transfer the culture to galactose, this is exactly the same experiment, 3 hours in galactose, we switch the number of cells expressing sense or antisense. So the number of cells expressing sense goes down, and the number of cells expressing antisense goes up. And these are genes which are expressed. Well, the sense transcript peaks in the oxidative phase, and the antisense transcript peaks in the reductive charging phase. So the take-home from this message here is that the reciprocal shift in transcription of HMS2 sense and antisense in cells grown in glucose versus galactose reflects the number of cells that either occupy the oxidative phase or the reductive phase, rather than a change in transcript levels in those cells. So when the gene transcripts are changing here, you're not seeing a change in the level of transcript in individual cells, you're seeing a change in the number of cells in the population that contain the transcript. The levels of transcript in these populations tends to remain fairly similar. And this, I think, is relevant to the question of what's going on with respect to the way trimethyl K4 is regulated. So let me try and summarize this a little bit. So in exponentially growing cells, if we shift the carbon source from glucose to galactose, we get an apparent repression of genes involved in growth and synthesis, and an apparent induction of the environmental stress response genes, the quiescent synthesis genes. I've nearly finished. This may simply reflect a change in the number of cells in the population expressing growth or ESR genes. So that's our interpretation. It's just a change in the number of cells. So if you go back to the question of why do GCN5 mutants lose trimethyl K4, which goes back right to the beginning. So I showed you that GCN5 mutants lose trimethyl K4, and this has always been ascribed to the role for GCN5 in acetylating lysine 14, because lysine 14A substitutions also lose trimethyl K4. But there's another alternative suggestion. So the proportion of cells in the oxidative phase of the YMC in a population of yeast is related to growth rate and nutrient availability. Many mutant strains in yeast have what's called generally a slow growth phenotype. So they grow very slowly. And this is probably relevant to you people in working with mammalian cells when you put RNAi onto a cell line to knock down function and the cells stop growing, because this is effectively the same sort of thing that we're doing. So mutant strains have a slow growth phenotype, and they have a higher proportion of cells in the quiescent phase of the metabolic cycle. So these genes, the genes that are expressed in the quiescent phase anyway, tend to have lower levels of Trimethyl K4 than the growth-regulated genes, explaining the apparent global decrease in levels. So there's nothing functional going on here. It's simply a shift in the proportion of cells in the population in one growth phase compared to another as a result of actually creating this gene knockout. So this explains partly why enzymes that deposit acetylation were believed to be required for Trimethyl K4, but we think this is an indirect effect as a result of change in growth rate. And there's a paper come out just at the end of last year from David Botstein's lab coming to exactly the same conclusion. So it's an important message for people working with gene knockouts, that when you make a gene knockout, if you affect growth rate, you're bound to be affecting this sort of biology, which is really important to keep levels normal. So many of the effects that people see are indirect. Right, one final point. If I've got, how much time have I got left? 2 minutes, okay, one final point. So this is another final point. And this is about the number of cells in a population and what they're doing. So this is a gene here called FO84. Same argument applies to our favourite gene, HMS2 here. This is data that I've taken from a paper from Daniel Zenklusen and Francois Stutz, because it illustrates beautifully the point. So if you look at the levels of steady-state RNA or the net-seq profiles, you would think that FO84 is a very highly expressed gene. Yep. And it's got very low levels of antisense transcription and antisense transcript. In fact, this is a cut, but still, it's still low levels of transcription here. So here we're measuring transcription. If you do RNA fish on this population, only 15% of these cells contain the sense transcript. This is the green blobs here. Each of those cells is stuffed full of transcript, all this stuff here. 25% of the cells contain an antisense transcript from this very low level of expression here, and about 60% of the cells contain no sense or no antisense. Now, the reason why this is important is because antisense transcription, even at very low levels, can affect chromatin. I'm going to summarize a lot of work here from Stuart Murray and my group. We found that by bioinformatics analysis that genes with antisense transcription in the vicinity of their promoter change their chromatin. They tend to have increased histone exchange, which leads to increased histone acetylation. So all these red nucleosomes here are acetylated. Decreased histone methylation, so decreased 36, 79, and trimethyl K4, and increased nucleosome occupancy at the promoter. So you don't really need to know that, but you just need to know that the chromatin, at least in yeast, associated with genes making sense transcript and antisense transcript, is different. So if we go back to the FO84 model, when you do your ChIP-seq on a gene like FO84, and you look at what the chromatin structure here is, most of the chromatin structure is going to be what is represented by what antisense transcription is doing to this gene, not sense transcription. And in fact, FO84, like many of these regulated genes with antisense, has a closed promoter conformation with high levels of acetylation, because most of the chromatin in the population, the number of cells with antisense here, is determined by the antisense, and only 15% of your population, in other words, 15% of your nucleosome prep or 15% of your chip, will represent the sense transcription profile. So this is my final message, if you like, which is to be very careful about the way experiments are interpreted based on what looks like a high level of sense transcription. Often it belies a very different distribution of transcripts in a population, and therefore the way you would interpret ChIP-seq data. So this is my last summary slide, and I'll finish here. So the last part of the talk, I haven't really shown you. I've very briefly mentioned that antisense transcription, which is very common in yeast and present at low levels, tends to create a dynamic chromatin environment with high nucleosome occupancy at the promoter and early coding region. And very low levels of antisense transcription, maybe 1 round per cell, is sufficient to bring about these changes. And analysis of chromatin structures, your ChIP-seq, ChIP-qPCR experiments will reflect the distribution of transcripts in a population. And the example with FO84, which has a closed promoter chromatin structure but transcriptionally resembles a highly expressed gene, reflects the fact that only a small proportion of the cells in the population are actually in the sense state. And that's my final message, and thank you very much for your attention.
Use this mic. So I'll start with a quick question. Getting back to the proline isomerization. So great data there from in vitro, but how much of all of that's relating to what's going on in cells where we don't have these mutants normally? There's another player, the Proline isomerase.
Yep.
How does that figure into your model?
Well, the Proline isomerases only increase the rate of transition. So the prolines will always isomerize, particularly in an unstructured region of protein. They'll always show a natural propensity to isomerize. Okay. So the tails isomerize. I mean, I think there's no doubt about it. It's very difficult to do this experiment. There is another paper just came out in Cell last year in mammalian cells where they'd attempted to do a similar sort of experiment by substituting prolines to raise antibodies to show cis and trans conformations. And they had similar sort of data to us. They were not looking at a histone tail, they were looking at another protein. But they were able to show using antibodies different states. So we think, I mean, we can only use the substitutions and draw correlative data, but if you put the two stories together, it makes a pretty persuasive argument, I hope. I've managed to persuade you.
Absolutely.
Very difficult experiments to do in the cell.
Great questions for Jane. We'll take Julie first as a microphone.
So, your ideas about the metabolic cycle and when you have slow growth mutants, you might get fooled. I'm wondering if you've tried to, is there a way to synchronize cells? You can test that idea, say in mutants that have, say, low overall levels, but if you synchronize them, you'll see that they'll have a normal level of, say, H3K4ME3 at the appropriate time.
We haven't done those experiments. Getting yeast synchronized in the metabolic cycle is not altogether trivial. But the TU lab have done some of those experiments, and we know that acetylation cycles and methylation levels in a normal cycle actually don't change, but we can fix our cells. So no, we haven't done that experiment, and I think it's a good one. It's a good one to try because that would be the prediction that you would see changes, and you know. You would expect to see in cells in the reductive charging phase lower overall levels of trimethyl K4 at individual genes, but maybe the population level would be the same because there's many more genes expressed there than expressed in the reductive, in the oxidative phase. So it's always this balance. So it's interpretation, but good experiment, yeah.
Tara and Jeta at the back.
My question is in context of effect of mutants on levels of H3K4 trimethylation. Could it be possible actually that these mutants are affecting the histone turnover rate? So the genes which actually retain the K4 trimethylation actually turn over less?
Well, that's a really interesting idea. I should have said actually that we know that none of these substitutions affect growth. I didn't put that data on. I should have done actually because there's no effect on growth. But yes, they could absolutely be affecting turnover, and it could be through lack of antisense transcription because all these genes tend to have their promoters protected by NRD1 dependent termination from antisense either coming from the 3' region or from non-coding transcripts trying to read into the promoter. And we would suggest that would mean they would be subject to less turnover and therefore be able to maintain higher levels. So we don't know the answer to actually why that class of genes behave differently yet, but that's a really nice idea, and we're thinking along those lines.
Nick?
So if I understand your idea correctly, the idea that depending on the structure of your histone tail will affect binding of, say, for example, bromodomains.
Yes.
But presumably the process of bromodomain binding will also change the equilibrium of the isomerization, because
Possibly.
essentially will be stabilizing
Possibly.
one state rather than the other state.
Yeah.
So I wonder if it's quite difficult thinking about the sort of the isomerization in isolation, because it'll be affected by the kinetics of the bromodomain binding.
Absolutely. I mean, that's the whole. That's the point in a way, that it's the dynamic structure, that everything is changing, and you can sway it in one direction or the other direction. And we don't know whether that's happening, but I would propose that a protein binding could capture a particular state, be it the cis state or the trans state. And as long as that protein is bound, you might be able to capture that state and maintain it. If the protein comes off, then you have the chance to change state.
So I guess the sort of follow-on from that is, if you were to do your chip with your isomer-specific antibodies, you'd expect different antibodies depending on whether you had a wild-type bromodomain or maybe a mutant bromodomain that couldn't then change the equilibrium of that site.
Yeah, we'd love to be able to do that experiment. We haven't done that. I think it's a great idea. We're a long way off from doing that. We do have bromodomain mutants which don't bind, but we haven't done anything in vivo with the SPD7 bromodomain mutants yet, mainly because they're also quite sick, unfortunately. And then you've got the problems with the effects on growth rate, but it's a great idea.
Any other questions for Jane? If not, thank you very much.
Okay.
Very provocative talk, thank you.